in Benavides from Silver River Museum. Alan, you're muted.
I muted anyway, my name's Alan Ivory, and I work with the Florida Museum for a program called Scientist in Every Florida School. And
we are so happy to bring you this episode of Fact or Fiction, where we have a claim and we're trying to provide evidence as to whether the claim is factual or fiction. Alan, you want to tell us about our claim for today?
Today's claim is all about scientists being know-it-alls. I've been called a know-it-all for many, many years, and I'm sure many scientists have heard that claim before. So today we're going to kind of review, is that factual or is it fiction?
And you have a video clip to show us, right?
I do, yes, I do. So the person I'm going to show you today for this video is someone who I thought was a know-it-all growing up, and maybe you have the same thoughts, but let's look at it.
Bill Nye the Science Guy. Bill Nye the Science Guy. Bill.
Did you know that white light is a mixture of all the colors of the rainbow? Red light can make you stop. Green light can make you go. The other colors can make you disco. Measuring allows us to make things that fit, and we can understand the world. Volcanoes are formed when hot, molten rock from deep inside the earth finds its way to the surface.
This is a liquid. This is a gas. And this, well, this is a solid. Now, we use these gauges and instruments to keep track of things we need to know, like how fast we're going, how high we are, how much fuel we have left. That way, we can measure things and land and fly planes safely every time.
Matter. Ever wonder what the universe is made of? Well, the universe is made of matter. And matter is stuff. Everything that you can touch is matter.
6 legs and flies. No. Insects. Insects are cool. Bees are insects. Of course, when you're around bees, you gotta be careful. Get it? Be careful. Ow!
So Bill Nye talks about everything from bees to volcanoes to properties of matter. He even flew a plane. So to me, as growing up, he just knew it all.
He really is a know-it-all. But are all scientists know-it-alls, or is that just something we think of because of pop culture, what we see on TV and in the movies? So for that, we have a special guest today, don't we, Alan?
Yes, we do. Today's special guest is Adam, and he's a PhD student with the University of Florida. That means he's trying to become a doctor.
But I almost forgot. Before we go to our special guest, I want to show you guys today's handout, just in case you have it in front of you, so we can talk about what you're going to do on it. Here's what your handout might look like if your teacher printed one out for you. So we have our claim here, and today we're going to be looking to fill in what Adam does do. So as a scientist, what can he do? What does he not do? And reasons why scientists might work together. And then after the Zoom, you might have an opportunity to do some of these critical thinking questions. So you might think about how a scientist collects their evidence to make sure it's good evidence. All right, evidence that we can count on. And then you might also think about why it would be difficult for a scientist to work alone to answer a question. And then, of course, you're going to either support our claim that scientists are know-it-alls or have evidence that refutes it. All right, so without further ado, let's bring Adam on board here. Hi, Adam, welcome.
Hi, how's it going?
Good. All right, Adam, before we get started with what you do, I have a couple of rapid fire questions for you. Okay, yeah. You ready? All right. Adam, what's the speed of light?
I don't know.
Okay, he doesn't know. What's a plutonic rock?
I'm gonna have to punt on that one if it's yes.
What's a tenric?
Don't know about that one either.
All right, guys. So I picked questions on purpose that I kind of had a feeling Adam wouldn't know. You're not a know-it-all, are you, Adam?
I am not, and I will never claim to be.
But you do know a lot about some things. So tell us what your science passion is. Who are you? What do you do, Adam?
So my name's Adam. I'm a PhD student here at the University of Florida, and I study aquatic ecosystems broadly, but for my research, I'm focusing more specifically on how manatees affect aquatic food webs and aquatic nutrient cycling, mostly through their poop and their pee. So I'm kind of the manatee poop guy, is what people refer to me as in the department.
So we have manatees in the Silver River. You mean they're part of the food web in the Silver River, right?
Yeah, exactly. Yeah, manatees are definitely a big part of the food web in the Silver River.
And even their poop is part of the food web?
Their poop can definitely be part of the food web, and that's what we're trying to look at with my research. More specifically, is does their poop function as a food resource for aquatic invertebrates?
But wait, a food resource, like things are going to eat it?
Yeah, definitely. Yeah, things might eat poop.
Okay,
you know,
so tell us how you would learn about that. What would you have to do to figure out if things are eating manatee poop?
Yeah, so there's a couple of different things you could do. You could go collect poop itself out in the wild, manatee poop, and see if you find invertebrates associated with it. And you could
have you done that? Have you gone out and collected their poop?
Yeah, I have. I've definitely gone out, and I actually have collected quite a bit of their poop, and I could show some to you right now if you'd be interested.
Yeah, absolutely. So do you just swim in the water? Do you have to put on scuba gear? What do you do?
Yeah, so for the most part, I just do snorkeling, but I do swim in the water with the manatees, and it's really fun to go out there during the winter months when they're in really high numbers, and you kind of have to fight with the manatees for space because they're so abundant in some of these areas. But yeah, we get in the water. We mostly do our work through snorkeling, but it's a lot of fun getting to swim around in the springs. But yeah,
right, show us one of these samples you've got and describe what you're looking at here.
So let's see. So here is what manatee poop actually looks like. This is, of course, dried. So manatee poop, it initially floats at the surface once the manatees poop, and then it eventually will get waterlogged and fall to the bottom of the water body, in this case, the springs. And yeah, it's about the size of a fist, and it looks very fibrous, and that's because the manatees eat predominantly seagrasses and freshwater grasses as well. And so it looks a lot like cow poop, if you guys have seen cow poop before. So it's very fibrous, and we're trying to see if manatees are actually providing a food resource for the aquatic invertebrates out there. So things like little micro shrimps, and there's also some crabs out there too. But we're trying to see if this functions as a food source for aquatic organisms, aquatic animals.
All right, so once you collect those samples, you've got those two in the lab. What do you do with those samples? Obviously, you're in a lab. What are you going to do with them to look for those tiny little critters, those invertebrates, animals without backbones that might be in the water?
Yeah, so what we do is we collect it, and then what we'll do is we'll rinse it off, and then we will put it in a greenhouse where nobody's working, so it can dry out, because it's really stinky actually, and you don't realize that until you take your mask off. So you're snorkeling around in the water, and then you don't know how stinky it is until you take your mask off. So we put it out to dry, and we rinse it off a little bit first, and we look for invertebrates that way. And we
just with your eyes? Can you see them with your eyes?
You can sometimes see them with your eyes, but oftentimes we need microscopes to look at them, because these are very tiny animals. We're talking about 1 millimeter to 5 millimeters in some cases. And so we use microscopes for a lot of our invertebrate work. And I've got just a picture of my setup here. We've got microscope and some fancy little lights that we can use to kind of concentrate the light on certain aspects of the little critters that we're looking for. But yeah, they're very tiny, and so we do have to use things such as microscopes to look at invertebrates and to really see who's there in our samples.
All right, so you showed us 2 samples. Is that all you need? You could just get a couple samples, look at them under the microscope, and you're ready to go? We're gonna have Dr. Adam soon?
No. Yeah, so it's still a long ways out for my PhD here. because there are so many samples that we have to go through and collect. These are just 2 samples that I have that kind of keep around, but we normally try to get at least 3 samples so we can get an idea of the true measurement, so we can get an idea of the average for any values that we're looking at.
Now, is that 3 for your whole experiment, or is that like 3 from one site?
Yeah, exactly. So it would be three for one site and for whatever specific variable that we're looking at. And so, for example, if we were to look at invertebrates at the springs, we would want to take 3 invertebrate samples where we use a little kind of metal cylinder, and we stick that into the sediment to collect the invertebrates. And so we want to always take at least 3 of these, what we call core samples, where we stick the cylinder into the sediments, because that gives us a better idea of the true average or the true value of invertebrates that are out at these specific sites. And so,
and how many different sites are you collecting these samples from?
Yeah, so I have it depends on what I'm doing, but I have 2 long-term study sites where I've been tracking manatees and their effects on the environment throughout the course of a year. And then I've got an additional 4 other sites that I've done some short-term monitoring. But what I've done was I have one site that has a lot of manatees there, and I have one site that has very few manatees that ever go there. And so I try to have 3 sites when I can for certain experiments that have really high numbers of manatees, and 3 sites that have really low numbers of manatees.
So it sounds like you've collected a lot of samples over time. I know in the first sim, you showed us a peek at just how many samples of whether they're core samples or what. Can you show us a sneak peek in your lab of where you're storing those samples and just how many you have?
Yeah, let me show you that right here. So we've got, so right here, we've got just a look at all of the shelves of samples that I've taken over the course of 1 year. And so these are all invertebrate samples that we've collected. And you can see this one, it looks a little bit pink. And that's because we put a dye into the sample, because it helps color the invertebrates and make them easier for us to pick out under the microscope. So again, we're talking very, very tiny invertebrates. We're talking usually on the size of 1 to about 5 millimeters at most. And so we've taken 3 samples from each of our 2 sites over the course of 1 year. And monthly.
So every time you go out, you're taking 3 samples. You're not just going out one time.
Yeah, exactly. I forgot to mention that I'm going out. I've gone out once a month for a year. And so I've got probably around 70 to 80 samples that I've collected over the course of.
Why so many? Why do you need so many samples, Adam?
Yeah, we have to get so many samples because replication and the number of samples that we collect is very important in order to know what the true value is. Because if we take one measurement of something, we don't know if that's representative of the rest of, if we were to take another sample out there. So we don't have a good idea of if one sample is representative of the true, if we were to take more samples, if that would change. Sorry, I'm kind of.
It's okay. So I think what I hear you saying is, in order for you to have what we're calling good evidence, you need a lot of samples.
Yeah, exactly. You have to have a lot of evidence to support whatever conclusions or assertions we are trying to make, because.
So why is your work important? Give us the sales pitch. Why, what does it mean for us?
Yeah, so my work's important because it's studying the effects of manatees on these ecosystems. And manatees haven't historically occurred up as far north as they have here in North Florida. And especially they haven't historically entered the springs in as high of numbers as they are currently. And so we're just trying to see what are the consequences and what happens when you have all of these manatees entering these freshwater habitats in the winter months. And so we're trying to see if manatees function similarly to Pacific salmon. And Pacific salmon, the eggs hatch in freshwater streams, and then the baby fish will swim out to the ocean, and they will grow up and spend the most of their life in the oceans, and then they will return to their native streams to spawn and to reproduce. And when the salmon enter these streams, once they're adults, they release a lot of nutrients, and they actually function as a food source for invertebrates in these Pacific streams. And so we're trying to see if manatees function similarly to Pacific salmon, in that their poop functions as a food resource for invertebrates, and that they might stimulate nutrient cycling and the uptake of nutrients in these ecosystems.
So I think that brings us into my next few questions, because we talked a lot about what you can do as a scientist, but we've already established you're not a know-it-all. So what are some of the things that are really important that you need to answer your question, but maybe you can't do? You have to turn to other scientists.
Yeah, so some things that you might need is learning new techniques. So that's something that we're always learning all the time as scientists, is coming out with new methods and new ways of measuring things and new ways of analyzing data. And so I have to oftentimes rely on other people to teach me new techniques or new methods of something or new ways to analyze data. And I'll have to admit here too that I'm not an expert in math. That's a subject that I've really struggled with over my lifetime. And so the level of math that I do for my work is pretty low level. And so I have to rely on other people to do some more complex calculations, or just double check over my work to make sure that I've done my calculations correctly. But in general, we rely on each other as scientists to teach each other new methods and new techniques. Kind
of like how you looked at the salmon scientist, and I imagine that inspired your work.
Yeah, exactly. Definitely the research that has come out of the studies from studying Pacific salmon has definitely influenced my work, and kind of gave me the whole ideas of, well, why hasn't anyone studied this already? So yeah, it's just always learning from other research and other scientists.
All right, so when you get ready to put all this together, you kind of do a big report, right? Almost like these kiddos are going to do a science fair project, only a little bigger, and other scientists are going to read it, right?
Yeah, exactly. Yeah, other people are going to read it, and so we got to make sure that we do the best job that we can. And
why is that important, that other scientists actually read your work and maybe even give you, well, I don't know, some comments and some feedback on it?
Yeah, so it's really important that we call it the peer review process. We review the work of other people's work, and that's kind of just how all science is done. We'll write up our findings in a paper at some point, and then we'll have to send it off to see if people agree with what we did and agree with our findings, because and that's kind of just to ensure that our science is sound, that the science that we're doing is sound, and that we're not just making stuff up, we're not making up our data, and that we're also not making conclusions that are overstating what we can actually say with our data, so that our data are sound and that our conclusions are reasonable based on the data that we've collected.
So you guys double and triple check each other as scientists, right?
Yeah, we do. We double check and definitely triple check. And it's a lot of work to get research published, is what we usually do with our results. We publish our results, but it's a lot of work, and it's very time consuming, and it undergoes a lot of opinions and critiques from other people in the field.
All right. Well, Alan, I think we're getting close to question time, right?
Right.
So I don't want to let anyone miss out on that. Alan, if you'll go ahead and spotlight yourself, because you're going to do Q&A, and teachers, you can start polling your students about what questions they might have and typing them in. And maybe Adam, while we're waiting for some questions to come in, is there anything really unexpected or surprising that's happened while you've been in the water with manatees?
I'm trying to think. Oh, I think the most interesting thing I've seen is actually manatees eating another manatee poop. So you might think of like dogs and cats maybe doing stuff like that. So, you know, I don't know what was happening that time, but I saw a manatee poop on the ground and I was looking at it, and then I saw another manatee come up and ate it. So, yeah, kind of an interesting thing that you'll see with like dogs and cats, maybe horses and stuff like that, bunnies. So it's kind of an interesting thing that I saw.
Now, how do people actually work in your lab? Both times we've seen you've been by yourself, but how full can your lab get?
Yeah, so we have a very tiny space and we have a lot of students working in here. We have a lot of graduate students. I think there's about 8 graduate students so far. And then we have probably about 5 to 8 technicians working in here. So we really don't have enough space for the work that we're doing, but we all try to be courteous of each other and try to let each other know when we're in the lab and whatnot. So we're not kind of taking up too much space and stepping on toes and whatnot. But yeah, we have a lot of people working on this project. There's probably about 2 or 3 helping me with my project, and then other helpers with everyone else's projects.
Oh, wow. Huh. This fields class selection note, do you ever notice a change in what the poop looks like? Just any changes with the poop, depending on the time of the year?
Yeah, so I can't really, yeah, it looks very different depending on, I think, how long it's probably been in the water before we collect it and whatnot. But in terms of the time of the year, that's not something we can necessarily see, at least notice with our own eyes, but we're going to run samples to see if the poop that is being ingested, that if the poop is mostly marine grasses versus freshwater grasses, to really kind of get at what are they eating. And we expect there to be more marine grasses when the manatees first enter the ecosystems in about November and December, and we expect that to switch more to freshwater grasses. But we don't have, there's not a way we can physically look at that and distinguish that, but there's some lab techniques that we're going to use to really be able to look at that and distinguish that.
I have never thought about the marine grasses versus freshwater grasses. It's pretty neat. Another question that we have is, how do you actually collect this poop? Are you just grabbing it by hand like you did here in the dried poop, or do you have specialized tools to do that?
Yeah, so to collect the poop, it kind of changes over time. Like when I first was collecting poop, I was really excited, so I just pick it up with my bare hands. I try to not do that anymore because it's kind of gross, and even using a lot of hand sanitizer can't really get rid of that smell super easily. So I kind of use just little bags, similar to like if you were picking up dog poop, if you were walking your dog or whatever. So I used to just pick it up with my hands, but now I just kind of pick it up with little plastic bags. That's usually how we store it, is we just put it in plastic bags and we freeze it.
That makes sense. In either the marine ecosystem or in the freshwater ecosystem, do the manatees face any sort of predators at all?
Do they face any predators? Yeah, they don't really have, manatees don't really have any predators. Yeah, they're one of the few organisms that has actually zero predators. So it's pretty interesting. Yeah, they don't have any natural predators.
Also, you talked about wearing like snorkeling gear before. Does this poop sink or float? Like, does that change what you use?
Yeah, so I think when it's fresh, it floats initially, and then after a little while, I don't know for how long, but I think it eventually does sink. And that's where I've collected most of my manatee poop is off the sediment underwater. So yeah, it usually, I think, floats at first, but then it will sink after a little while.
I found this question pretty interesting as well. Are the digestive systems of humans similar or different than that of manatees?
Yeah, so they're actually different. Let's see, I'm gonna mess this up. Another thing that I don't know at all, but I forget what the human gut is, but manatees are actually hindgut fermenters, and they have a very long digestive tract. And so it takes manatees 7 to 10 days to pass the grasses that they eat through their gut and to develop it, to poop it out. So manatees have a very long gut, and it takes them a long time to digest the food that they eat, which is different from humans.
So you'd say it's kind of similar to a cow, maybe?
It's more similar to a cow, but it is different though. Cows are ruminants, and manatees are hind gut fermenters. And I feel I feel bad. I can't remember what.
No, but the poops look similar. That's what I was gonna get at. Poops.
Yeah, they're pretty similar. Yeah.
Right. I can't not ask this question. How has COVID affected your research at all?
Yeah, so we've actually been pretty lucky in being able to not have too many issues with COVID affecting research. I know there's been other people throughout different states who have had different issues with COVID, but I've been pretty lucky with COVID, and it hasn't really interrupted my research that much. We had about 2 or 3 months, I think, when COVID was just becoming a big issue in the United States last year. Around March through about June, we didn't do any sampling, so we had to stop sampling and resume after that. But it hasn't taken too big of a toll on my research, which I'm very lucky and very fortunate about.
One last thing I want to mention, it's not very sciencey related, I guess, but what type of scientist would you claim that you are as a manatee scientist? Like, I claim to be an ecologist. What do you call yourself?
Yeah, so I also consider myself an ecologist. I study ecosystems, and that was kind of where my training was developed, was studying ecosystems and the effects of organisms on ecosystems. So I consider myself an ecologist who studies the effects of animals and the environment, and how the environment affects animals living in an ecosystem, for example.
All right. Adam, thank you so much for joining us today. We have had a great time learning about how you work as a scientist. And students in class, I hope this information has helped you understand a little bit more about science and scientists. And hopefully you have some good evidence to think about that claim that scientists are know-it-alls. And so I hope you guys will join us again in a couple more weeks. And Adam, thank you again. All right. Bye, everyone.